Hello everyone and welcome to this screencast about evolution and the mutation spectrum. The focus is going to be on how the way that variation happens can shape the way that evolution happens. And my focus here is going to be on theoretical possibilities. What does theory tell us? Gave a background presentation covering this other issue of where do variational tendencies come from. Some key takeaways from that are that a mutation spectrum, when I say the word mutation spectrum, that's any way of breaking a mutation rate down into component parts. And for purposes of causation and models of causation, for at least some context, it doesn't really matter if we're talking about a bias toward one phenotype relative to another phenotype, one RNA fold relative to another RNA fold, or one molecular character or nucleotide relative to another nucleotide. Those are all different versions of the idea of a bias in mutational accessibility. And then finally, if you're thinking about a research program where we call on variability in mutation rates or heterogeneity in mutation rates to explain things about evolution, the inputs to that are differences in mutation, and mutation rates vary in just about every way that you can imagine. Okay, so with that background, I'm going to cover a lot of different topics about how the way that mutation or variation happens can affect how evolution happens, and to organize this, I'm going to give you three different ways of thinking about how to organize this material. One of them is, what's the role being played by mutation? Is mutation the load or pressure of mutational degradation? Is it a source of raw materials that selection shapes into adaptations? Or does variation, is it represented as events that introduce new things? The second dimension for organizing this knowledge, what we know about how mutation can affect evolution, is how much of the complexity of mutation is involved. So in classical theory, if there's a mutation rate at all, it's usually just one number, a scalar value for the mutation rate that's intended to represent all mutations as the same thing. A more complex way is to have multiple different rates, to break the mutation rate down into some kind of a spectrum, and to allow the mutation rates to vary according to other factors in the model, like environmental conditions. And then finally, how much of the complexity of evolutionary dynamics are involved in our theory about how mutations affect evolution? Is it just the immediate effect, or is it something that's more long-term, or even something that has indirect effects and feedback effects? So I'll start with this idea of mutation as degradation. So in these kinds of theories, mutation has the effect of degrading a trait, including fitness or some phenotypic trait, and then that has some evolutionary impacts. So the most familiar kind of model from classical population genetics is something that Fisher and Haldane developed to understand why we have genetic diseases, because there's selection against things like hemophilia. Why don't they just disappear from the population? And the answer is recurrent mutation pushes up the frequency and selection pushes down the frequency. And in a sufficiently large population, then the frequency reaches an equilibrium or balance point. That's called the mutation selection balance. And because mutation rates are small and selection against diseases is strong, that equilibrium frequency is a small number, much closer to 0 than to 1. Another important idea historically is evolution by mutation pressure, and this is something that's not very well understood today, but this had a clear meaning to Fisher and Haldane, which was the mass conversion of a population by mutation, and it's shown very well in this simulation, Evolvulator simulator from John McDonald's website. So this represents a population here with about 20 individuals in each generation. Each generation is a column, each row is a lineage. So each individual in a particular generation has one offspring in the next generation. So there's no selection and no drift that way. The only thing that can change the population is mutation. And here, the mutation rate from the red allele to the blue allele is set to be very high. And so over a relatively small number of generations, there's just 100 of them shown here, the population is converted from red to blue. Okay, so this is not like the way we usually think about evolution by reproductive replacement. This is conversion by mutation. And it's usually, and this kind of evolution, evolution by mutation pressure, is thought to be generally unimportant because you'd have to have high mutation rates unopposed by selection. But the kind of case where it might be useful or applicable is in something like the loss of an unused trait, like eyes in cavefish, for instance. So here's an example of a contemporary analysis of the loss of a complex trait, sporulation in B. subtilis, and this paper by Mon et al. Has both some theory and some experiments showing the loss of sporulation by mutation pressure and drift. Another application of this idea of mutation pressure is adaptive reduction of the mutation rate. So from load theory, deleterious mutation is the same thing in essence as a fitness cost. And the cost is proportional to the mutation rate, and under some conditions you don't need to know what the exact distribution is. And the reason for that basically is that a lethal mutation is lost in one generation. It's like one selective death. Something that's not as lethal, just causes a partial reduction in fitness, will be spread out over multiple, the cost will be paid over multiple generations in multiple partial deaths. Okay, so given this idea that deleterious mutation entails a cost, if you reduce the mutation rate, if there's some genetic modifier that reduces the mutation rate, then that's potentially selectable with the selection coefficient that's proportional to the reduction. And what's the important thing to determine is that selection coefficient big enough relative to the inverse of the population size, big enough to be selectable. Okay, so those are some of the basic theories about mutation and mutation pressure. What do we get when we take this idea of mutation as degradation and add the idea of different rates to it, to the load theory, or to loss by mutation pressure, or to mutation rate evolution? Well, here's an interesting idea from this paper by Berg and Kurland, why mitochondrial genes are most often found in nuclei. And what they argue is that if there's some process that gets us from this state over here, where we have a gene in the mitochondrion but not in the nucleus, to a gene in the nucleus but not the mitochondrion. And by the way, the background for this is that there's a long-term evolutionary process of transfer of mitochondrial genes to the nucleus, so that in animals, for instance, the mitochondrial genomes are very, very small. They only have dozens of genes, whereas they started out billions of years ago with a full complement of a bacterial genome with thousands of genes. So what Berg and Kurland argued was that, well, if this process is happening, there must be some intermediate where there's a working copy in the mitochondrion and in the nucleus, and then the question becomes, which one's going to get lost? And what they argue is, well, the mutation rate is much higher in the mitochondrion. And so that's the copy that's going to get lost. That is, this intermediate where you have both of them will be resolved by the loss of the mitochondrial copy because mutation pressure is higher. And you could add that same kind of thinking, you could apply that same kind of thinking in other cases. So for instance, we would expect Y chromosome genes to get lost because the mutation rate is higher there. And in the case of the loss of traits that I mentioned previously, the more complex the trait is, so sporulation is a complex trait in the sense that it's encoded by many genes. It has a large target for mutational degradation, and the larger the target, the more complex the trait, the more susceptible it is to this kind of loss by mutation pressure. As another example, just thinking through and applying these concepts, CPG hotspots, CPG dinucleotides, are highly mutable in animals and mammals and birds. It's because they're methylated. There's about a 10-fold effect on the rate of transitions, several-fold effect on the rate of transversions, and it's a frequent contributor to human disease. In fact, very early on in the identification of disease-causing mutations, it was clear that CPG mutations were making a profound contribution to monogenic diseases. And this raises a really interesting question about why. So if a site like a CPG site is a hotspot, if it's mutable, then one predictable aspect of its long-term dynamics is that it's going to get lost because it's easy to lose. It's losable at a higher rate. And in fact, CPG sites in the human genome are about 8-fold lower than what you'd expect from the frequencies of C and G. Now, what's going to happen to the distribution of fitness effects of mutations at CPG sites as a result of this process of loss? Well, they might start out with some particular distribution, but over time, the CPG sites that will disappear are the ones that are easy to lose because they don't have strongly deleterious effects. So what's going to happen over time, over many hundreds of millions of years, as CPG sites are lost, is that the remaining ones will be enriched for sites that have highly deleterious effects. So for instance, if a site encodes an essential, a CG could be an arginine codon, a CG and arginine codon. All right, if that's an essential arginine in a protein, then it can't get lost, and that's the kind of CG site that will stick around, whereas other ones that are in intergenic regions, for instance, can mutate away. And possibly that is the explanation for why CPG sites contribute so much to disease. And it has practical implications, like this is actually a strategy for making adoptive T-cell therapy, is to target hotspot mutations like R175H in the TP53 gene, okay? Because if you're going to design an antibody therapy that will work on different kinds of cancer, you want it to work in different patients, and different patients typically have different mutations, except for these hotspot mutations, which are very, very common. Okay, another implication that we get by starting with this idea of mutation as degradation and then adding in the idea of different rates or a spectrum of rates is targeted hypomutation. So to begin with, classical thinking always held that targeted hypomutation, the idea that mutation would be depressed in important genes or at important sites, that was often argued to be infeasible due to that requirement I mentioned earlier of the selection coefficient being large enough. I've got my, this should say larger than rather than less than. But in 2013, Inigo Martin-Carena and colleagues made this argument that, well, that's if a modifier only if the modifier that reduces mutation just does it sort of randomly all over the whole genome. But repair pathways and mutation pathways are affected by the structure of the chromatin and what binds to the DNA. And there's lots of signals, there's lots of aspects of the structure of the genome and chromatin that have associations that differ for genes and non-genes. What if a repair mutant, some kind of modifier of the mutation rate, tended to reduce the mutation rate in genes versus non-genes, not because they're genes versus non-genes, but because they have particular chromatin marks on them. And what they argued is that by making this kind of association of repair with transcription or chromatin-associated proteins, they could overcome this problem of the drift limit and lead to what they called non-random hypomutation, meaning like a correlation between, an anti-correlation between the rate of mutation and functional density, so that the more functionally dense regions had lower rates of mutation. And this is something that we heard about in the very first talk in our series when Gray Monroe talked about targeted hypomutation in Arabidopsis. Okay, so to summarize, we begin with this basic idea of mutation as a pressure of degradation and add the idea that there's a mutation spectrum, and what we get are some different theories where the nature of the mutation spectrum influences how evolution turns out, influences how diseases are generated, influences which traits tend to get lost, influences how genes get transferred from one compartment to another, and may produce patterns like targeted hypomutation. All right, now let's go on to the second conception of the role of mutation in evolution, which is a source of raw materials. And this comes pretty much straight from Darwin's thinking. So Darwin offered this analogy of a builder building something from uncut stones that fall from a cliff. I like to think of a slightly different analogy of making a sandcastle out of some sand. So because it's very clear here that the sand is just providing raw materials. It's selection in the form of the mold here that creates the shape. And that's in spite of the fact that every individual grain of sand, just like every variation, is unique and has a unique history. Those are irrelevant details if we're trying to explain the shape of the sandcastle. That shape is due to selection, and the role of variation is just to supply raw materials. Now, an interesting thing has happened to this sort of line of argument, which is that when you make formal theories, when you make formal models, mathematical or computational models to explore a theory, sometimes they get out of hand and they do things that undermine the theory, and that's what's happened in this case. So what I mean, but let me just give you an example of what I mean by this. So here we have a model of population growth that's called the logistic growth equation, and it was used to argue that populations regulate their growth. So if we start out with a field, that doesn't have any mice in it because there's been a flood or there's been a fire or something like that, and over time it will get repopulated, the population will grow and grow, but as it nears the carrying capacity, growth will slow down and then it will level off. Now, so here this model exemplifies the theory that populations regulate their density. But if we fiddle with this kind of rebound parameter here, r, and we make it a little bit higher, then the population at first will overshoot the mark, and then there'll be a dampened oscillation. So that sort of still suggests this theory that populations regulate their density. But if we turn it up even further, we get this weird periodic behavior where the population just keeps jumping back and forth between 2 or 4 or 8 different sizes. And if we go even a little bit further than that, we get deterministic chaos. Okay, so exactly the same equation, we change the parameters a little bit, and it used to be a model of how populations regulate their density, and now it's a model of something else. It's showing a different kind of behavior that wasn't anticipated at all by this theory. Okay, here it's a model of deterministic chaos. So what happened with this raw materials way of thinking is that based on first on intuition and then based on things like Fisher's fundamental theorem or the Breeder's equation, advocates of neo-Darwinism argued for generations that if selection just has a little bit of variability, then it will take the population to the optimum. All right, so as long as there's a little bit of variability in every trait varies up and down just a little bit, then selection can do whatever it wants. But when the mathematics of multivariate quantitative genetics was worked out formally by Landon Arnold, the result actually said something different than that. What it said was that the expected changes in the trait values here, these delta z's, is actually the product of a matrix, a G matrix of variances and covariances and selection and a vector selection differential. So what this means is that the change I expect in trait 1 is not just dependent on multiplying this vector of this selection differential with the variability of trait 1, it also depends on selection on trait 2 and 3, how they act on traits 2 and 3 has an indirect effect called indirect selection on trait 1 based on the covariance of trait 1 with trait 2 or the covariance of trait 1 with trait 3. All right? And so on that basis, in this review, Stepan and all, about the G matrix, say that together with natural selection, the G matrix determines the direction and rate of evolution. So what the theory says is that selection and G jointly determine the direction and rate of evolution. It's not one or the other, it's both of them jointly. And this was expressed initially in terms of the G matrix rather than mutation. G matrix is standing variation, but we can push this back to mutation by saying that there's an M matrix that represents the contribution of new mutations to trait variability, and argue that if G is shaped by M, then G can shape so R here represents a matrix of divergence. Let's say we look at a bunch of different species that are diverging under the same G and beta, they will show some dimensions of divergence that we can represent as a matrix R. Now, that's a very technical way of explaining what this theory, the multivariate theory says, but it's usually expressed more relative to Darwinism as genetic constraints. Okay, and that was first stated pretty clearly in this paper from Schluter. So what he argued was that, suppose we're starting out with a population like this. This is what variability for two traits looks like. Here's the optimum over here. What happens in this case is not that the population doesn't just go directly to the optimum. It tends to go in the direction, tends to start out initially in the direction of maximum variability. And the way to understand this is just that even if the population is sort of maximizing the increase in fitness, sometimes you increase fitness faster by going rapidly up a shallow slope instead of going slowly up a steep slope. All right, you can go rapidly up this shallow slope if there's more variability here to fuel change. But the way that this is conceptualized, the way that this is presented is a constraint. It's kind of a temporary deviation of the path of evolution from this optimum that over time, so over longer and longer periods of time, in Schluter's conception here, selection is eventually going to take the population to the optimum because there's some variability in every trait. Okay, so over the years there's been empirical results that support this idea that there's a correlation between G and R, and more recently between M and G and R. So here's an example from this paper by Hull and colleagues from 2017 where they're showing M, G, and R for a bunch of different wing traits. And in this paper, they didn't come to a firm conclusion. One of the sources of ambiguity was that although you could get an M-R correlation because M shapes R through G. That's the constraint hypothesis. It's also a theoretical possibility that recurrent correlational selection can actually shape the M matrix indirectly over time. And so you could get a correlation between M and R because of selection shaping M and selection shaping R, emphasizing the same dimensions. I won't say much more about that except that in this very recent paper by Holstadt and colleagues claims to resolve this issue in favor of the constraints hypothesis. That is, they think that the most reasonable explanation over a large set of data is that there's an M to R correlation and it's due to M shaping R. Okay, so to summarize the theory, there's this canonical multivariate theory that says that the rate and direction of evolution are jointly determined by G and selection. And this led to this constraint hypothesis, but there's another possibility of correlational selection shaping N, and there's some other results that I'll talk about a little bit later after we've covered some more other theory. Okay, and then finally, the third way of thinking about the role of mutation in evolution is in terms of events that introduce new things. So over here on the left, we have a population of these blue crabs, and there's an event of introduction that introduces a slightly different yellowish colored one here. And then it's possible over time for that to go to fixation by reproductive replacement, by selection or drift. This way of thinking was formalized in 1969 in origin fixation models, which represent the rate of evolution as the product of a rate of introduction and a probability of fixation. And we've all probably seen two different versions of this. There's the neutral version, where the rate of evolution is equal to the rate of neutral mutation, because the 2's and the n's cancel out here. And the adaptive version, where the rate of beneficial changes is equal to 4 times n times the rate of beneficial mutation times the selection coefficient of the beneficial mutation. And one of the implications of this way of thinking is that if there's an evolutionary process that depends on events of mutation that introduce new alleles, then it's subject to biases in the introduction of new alleles by a kind of first come, first serve effect. This was first shown in 2001 by a paper that I did with Levy and Polsky. So in this model, we basically have two options. They're both upward steps. Like it's a one-step adaptive walk. You can go to the left or you can go to the right. And one of the options is accessible by a greater mutation rate. The other one has a greater fitness benefit. And the question is, how often will you go to the mutationally favored one as a function of mutation bias? And the results shown here provide an answer to that. So regardless of the population size, the more you increase the mutation bias, the more you see an effect on the bias in outcomes, going to the left versus going to the right. And under conditions where mu times n is small, the behavior of these are computer simulations. The behavior of the population corresponds to what we expect theoretically for origin fixation dynamics, where the rate of evolution directly reflects the bias in mutation. And this idea of arrival bias has manifold implications. So if there's an evolutionary preference for mutationally favored options, that could create a tendency for parallelism. It could account for taxon-specific or clade-specific propensities of change, and a bunch of other things some of which I'll explain briefly here. So in terms of parallelism, suppose that we have a bunch of different possibilities that might happen, and let's assume these are adaptive possibilities. There's a dozen or so of them here, and we'll represent them on a number line because each of them has some kind of probability. The chance that we'll see on two different lineages that we'll see the same event is then a sum of squares over these elemental probabilities. And we can also express that in terms of the variance or the coefficient of variation, and this shows us that if the variance is 0, if all of the possibilities are equal, then the chance of parallelism is just 1 over n, where n's the number of possibilities. But it also tells us that the greater the variance, the greater the chance of parallelism. Now, I just mentioned that under origin fixation conditions, the chance of something happening reflects both the mutation rate and the selection coefficient. And we can actually express that, at least under origin fixation conditions, in this way, that the chance of parallel evolution reflects these two terms here that are based on the variability of the mutation rates and the variability of the chances of fixation. I don't have the reference here, but there's a paper by Gitschlag that will be in the bibliography a little bit later, Gitschlag et al. 2023, that gives some examples of using this equation on actual data. So in order to determine what fraction of parallelism is due to heterogeneity in selection coefficients, what fraction is due to heterogeneity in mutation rates. Arrival bias can also contribute to directional trends. This was shown in a 2006 paper here that looked at the effect of GCAT biases on protein composition in adaptive walks. So these curves here represent the average of many adaptive walks, each of which starts at a random position and goes to some peak, either under high AT bias or under high GC bias. And under high AT bias, the proteins tend to accumulate the amino acids that have AT-rich codons, and under the opposite bias, they tend to accumulate the amino acids that have GC-rich codons. So you get this trend in amino acid composition that reflects the mutation bias in adaptive walks. And it's important if you want to understand this, it's important to toss out the idea that evolution is some kind of zero-sum game where either selection wins or mutation wins. If adaptation is happening in a multidimensional space, then there are many different ways to go up. And evolution go up in fitness, go up in adaptation. And evolution tends to go up in directions that are mutationally favorable. That's the way to understand how both effects can happen at the same time. All right, another effect here was shown in this paper by Kano and Payne. They took empirical fitness landscapes for transcription factor binding sites. And for each one, they identified what's the major peak on this for this transcription factor binding site, and from all the possible sequences, what are all the paths up to that peak? And then they asked, summing over all those paths to the peak, how many transitions are there and how many transversions are there? All right, and usually the ratio of transitions to transversions is 0.5 to 1. Okay, but there's variability. And what they showed was that in evolution, the chance of reaching a peak is highest when the mutation bias of the evolving system matches the composition bias of the landscape. So they varied this transition-transversion bias, and they looked at different landscapes, and what they found is the chance of reaching the peak, what they called the navigability of the landscape, is highest when the mutation bias matches the composition bias. Okay, another extremely important idea that we've heard of in our seminar series this year is this idea of self-organization by the arrival of the frequent. So here we have on the left here, this is a genotype-phenotype map for RNA folds. So each dot is an RNA sequence or a genotype, and the color represents the phenotype. If the sequence folds up into this form, it's colored in blue. If the sequence folds up into this form, it's colored in gray. And if the sequence folds into this form, it's colored in red. And there's a difference in accessibility. If you're starting out in the blue network, all right, the chance of a one nucleotide mutation path into the gray genotypic network is considerably higher than it is for going into the red network. All right, so there's a bias in accessibility, and this has the effect of putting a prior bias on evolution due to arrival bias. You're more likely, if you're starting out in the blue phenotype, to go to gray than to red. And one of the implications of this, given the properties of genotype-phenotype landscapes, is that, so in genotype, it's a general property of genotype-phenotype landscapes, of various genotype-phenotype landscapes, that there are a small number of very, very highly frequent phenotypes that have many, many, many genotypes. And they tend to dominate the genotype-phenotype map, and they tend to be the most likely in evolution because they are more mutationally accessible. So the findability of these very common phenotypes is due to an effect of mutational accessibility. Okay, one last idea here. Oh, actually, there's one more after this. Has to do with bias reversals. This is another topic that we've heard about in our seminar series this year. So during an adaptive walk on a finite landscape, there's some number of possible beneficial changes. And as adaptive steps happen and beneficial mutations are fixed, possibilities are removed from that set of beneficial mutations. All right? So the total set of beneficial possibilities remaining to be explored gets smaller and smaller. And if evolution is happening in something like origin fixation conditions or in mutation-limited conditions, then that's going to be subject to mutation biases. And what this graph shows is that the distribution of fitness effects, so this is looking at the beneficial fraction, is depleted more strongly for transitions than for transversions because the transitions are happening at a higher rate. All right? And one of the implications of this is that it becomes a possibility then that over long periods of time, adapting lineages will undergo bias reversals, because if you've gone partway through an adaptive walk, all right, flipping the mutation bias can change, can increase the chance of adaptation by accessing possibilities more frequently that used to be less frequent. Okay, one final point. This is my final point here about this way of thinking has to do with the theory of mutators. So the classic theory of mutators was often used to argue that selection is going to keep mutation rates low. And yet it was observed again and again, for instance, in E. coli, that there are mutators in clinical isolates. So there's strains that have a mutation rate that's 10, 100, 1,000-fold higher than the typical mutation rate. And this led to theoretical exploration and simulations and experiments about mutators and anti-mutators. And the gist of the theory is that when the environment is highly variable, it can be beneficial to have a high mutation rate. But then as conditions stabilize, it becomes, or over time, then modifiers are favored because increasing the mutation rate increases the load. Okay, so this mature theory of mutators has both mutators and anti-mutators. Mutators that increase the mutation rate, they're favored in the short term under variable conditions, and then over the long term, modifiers will be favored that tend to reduce the mutation rate again. So this is not all that I could say about this, but I just want to make one point that has to do with specialized mutation systems. So normally we're told that mutation happens completely independent of what's beneficial and that there aren't any kind of specialized mutation systems. And that's just not true. The example that everybody knows about, we hear about in molecular biology class, is the maturation of antibody genes. So in this system, there's a whole library of different V segments, D segments, and J segments, and there's a programmed system of DNA rearrangements that makes a join between a D and a J, and then makes a join between a V and a D. And the result of this, it's like picking a card from each of these three decks and putting them together to make a functional gene, which is then actually subject to another process, which is hypermutation. All right. Now, this process has been known since the early 70s. But it's a somatic process. It doesn't change germline genes, so it doesn't contribute directly to evolution. However, the fact that this system exists means that there's no biological rule against specialized mutation systems existing, and there's no evolutionary rule that prevents them from coming into existence. And in fact, if we look at bacterial genetic systems, in particular pathogens, what we see is that there are a lot of different specialized mutation systems that enhance the rate of specific mutations that are used in adaptation, and they have specialized parts, they're regulated, and they've evolved repeatedly. Some of them are like the antibody maturation system in that they involve cassette donation systems. So this would be like trypanosome antigen shuffling. There's some that use inversions to create diversity. There's a bunch of different types of diversity-generating retroelements where diversity ends up emerges through a stage of error-prone reverse transcription. And there are hundreds and hundreds of separate examples of phase variation, either by this STR mechanism or by this inversion mechanism. And if you want to read about these systems, it's in chapter 5 of my book. Okay, so to summarize this third role of mutation as the introduction of novelty, this idea of arrival bias, again, has manifold implications. It leads to this idea of preferences, directional trends, self-organization, bias depletion, and bias shifting, navigability of landscapes, and so on. When we combine the basic idea with diversity in mutation rates and with long-term dynamics and feedback loops. Okay, now before concluding, I want to do just a little bit of analysis here. So I presented these different ways of thinking about the role of mutation and apply them to some of these different topics. And I want to talk a little bit about, well, how distinct are these different things? It's important to distinguish models from theories, first of all. You could write a simulation of the evolution of a quantitative trait, with some discrete number of loci and mutations with effect sizes and a population and so on. You could also make a model of the loss of a complex trait by mutation pressure, and you could make a loss of mutation-biased parallel molecular evolution. So you could make models in the three different classes using the same simulation software. And there are some theories that rely on multiple roles. So remember, the theory for mutators relies both on mutation, the load arguments that are based on deleterious mutation pressure, and it's based on the idea of mutators that arise because they are more likely to introduce a beneficial alternative. And I mentioned this gene transfer theory from Berg and Kurland, which made an argument based on mutation pressure, but actually, if you were to look into this further, you'd realize pretty quickly that there's also an effective introduction bias there too. So there's a pretty promiscuous process of transferring bits of mitochondrial DNA to the nucleus in animals. There's not such a promiscuous reverse process going in the other direction, and that surely has an impact on the dynamics of long-term transfer. But an important thing that I want to emphasize is that the ideas that have emerged in these different fields are not the same thing, and in particular, arrival bias in molecular evolutionary genetics and constraint in quantitative genetics are not the same thing. They are dispositional effects in a sense, but they operate by different rules, and if there's a bridge between them, I don't know what it is. I haven't seen it. So this constraint effect that I explained is dimensional but not directional in classical quantitative genetics because it squeezes everything down into variances, okay, that have a size but not a direction. So this effect here of this ellipse going in this direction is the combination of the ellipse and selection differentials going up toward the optimum here, right? But if the optimum could be someplace else, and it would be just as likely for this ellipse to, when combined with selection, point the population in the blue direction instead of the red direction. Okay, so again, the effect of variability within this conceptualization, within this formalization of quantitative genetics, is dimensional, not directional. And that's not really what evo-devo people are typically talking about. So for instance, Wallace Arthur is a leading thinker in evo-devo, and he draws pictures like, here's this embryo. This is just a thought diagram here of this embryo changing in some directions more than in others, and in some directions apparently is not able to change in that direction at all. Or this is another figure from Wallace Arthur's from his 2004 paper, where he's showing an effect of variability that he's labeling the M matrix here. But what's happening is that it causes the population to favor one peak instead of another peak, all right? This asymmetry of variability, which is not really what's happening in the quantitative genetics models, all right? Now, there are some quantitative genetics papers on directional mutation bias. So this paper by Waxman and Peck, and then there's follow-ups by Zhang and Hill and by Charlesworth. What a mutation bias means for them is that, like, considering a single trait, all right, mutations that affect the trait are more likely to increase the trait value than to decrease the trait value. That would be a mutation bias if you have some slight tendency for mutation to increase rather than decrease the trait value. Or the opposite bias, a tendency to decrease rather than increase the trait value. But in these models, there's just a single optimum. And so what happens is that without the mutation bias, selection takes the population to the optimum, where the mean phenotype is the optimal one. But under mutation bias, there's just a slight deviation from the optimum. Instead of the population resting on the optimum, it's just a little bit off. All right, which is not a very interesting effect. And again, it's not what people in evo-devo are talking about. And to be fair to Waxman and Peck, they know this. So they say this small effect of a bias in our model they're talking about depends on the existence of just one optimal phenotype, as assumed throughout. If multiple optima exist, then a small amount of bias may have a very large long-term effect. Okay, finally, I'm going to wrap up with a few conclusions here. So what I've been doing, I've been thinking about this for a long time. I'm trying to do a kind of synthetic review of all of this work on the role of mutation in evolution to assess the breadth and depth of published work, to relate different areas of the literature, explore some unifying concepts and languages, identify challenges and so on, and ultimately to think about the prospects for a structuralist research program. Okay, and I mentioned these different dimensions to structure our thinking, or to think about the literature. So what's the role of mutation in one of them? Now, I'm not saying that this is the best way to think about how evolution works, but it is a really good way to understand the literature and to understand how evolutionary biologists think and how they have thought in the past. Are they talking about novelty introduction? Are they talking about raw materials? Are they talking about mutational degradation? And it's also really important to think about the complexity of mutation. On a lot of classical work, there's just one mutation rate, okay? But now we know that mutation is heterogeneous and variable. And the effect of mutation can be different depending on whether we're talking about just a short-term effect, a long-term effect with feedback. Those can all create interesting dynamics. So one of the issues that has come up to me again and again is the need to bring the whole field of evolution up to date on what we know about the role of mutation in evolution. So one of the things I see again and again is that people who are writing about this, appear to be authorities on this, are getting things wrong. So just as an example, oftentimes all of these concepts here, like these five, what I would say are five different things, are being conflated. So mutation bias is a concept from genetics or genomics. It's a pattern where there's a systematic difference in mutation rates, all right? And we've known about mutation biases since the 1960s. If you look at the literature, transition transversion bias, GCAT bias, male mutation bias, those are the three most common types of mutation bias referenced in the literature. And there is not in any case an implication that the mutation biases are adaptive. Okay, they're not associated with targeted hypermutation or specialized mutation systems. All right, that's a different thing. Arrival bias is a theory in evolutionary genetics. Okay, mutation bias is a genetics concept. Arrival bias is an evolutionary concept. Okay, mutation biased adaptation is the empirical pattern that evokes arrival bias. It's when we look at adaptive changes and we see that the changes involved in adaptation tend to be the ones that are mutationally favored. And this is an empirical pattern that has only been established firmly in the last 5 years. So that's a very recent thing. Targeted hypomutation is the idea that there's an inverse correlation between mutation rate and functional density. More mutation in less important regions, less mutation in more important regions. And it's kind of been folklore for a long time. People have been saying this for a long time, but it wasn't really established based on clear it didn't become a credible empirical argument until very recently. And the theory for it didn't exist until that paper from Martin Carrena that I mentioned in 2013. And likewise, the idea of specialized mutation systems has mostly been folklore among molecular geneticists that hasn't been accepted by evolutionary biologists. Okay, anyway, so there's key findings that are not integrated into evolutionary thinking. One of them is that specialized mutation systems really do exist. Mutation bias adaptation is widespread, and arrival bias has a lot of implications. Another issue is to identify strategic gaps. So one of the strategic gaps that came out of the analysis that I've been doing is this lack of alignment between concepts in quantitative evolutionary genetics and concepts in molecular evolutionary genetics, and the need for some kind of theoretical alignment, which I would love to work on if I were more of a theoretician myself. Another one is, what's the nature of developmental bias? So as evolutionary biology as a field needs to have a discussion of what we mean by this and what would it mean to establish developmental bias. And we can't have that discussion if people are still confused about what kind of causation we're talking about. And are we talking about a dimensional effect, or does it have to be a directional effect? Self-organization is another one. So when Stuart Kauffman wrote his famous book in 1993, The Origins of Order, he said that order comes about through this combination of selection and self-organization. But at the time, it wasn't clear what he meant, and Stuart Kauffman did not know what he meant. He did not have a clear philosophical treatment of what is self-organization, what is selection, and how do the two of them work together. I would say that we know the answer to this question today, mostly due to work from Ard Louis' lab that I've talked about and that Nora Martin and Kalmadin Dingle talked about. A third issue, as I mentioned previously, is articulating a research program. So there is the potential in evolutionary biology for a research program that's focused on the dispositional role of variation generative biases in evolution. It would integrate key concerns of evo-devo and molecular evolution. It would contrast clearly with the historical neo-Darwinian functionalism. It can already lay claim to some distinctive and important theoretical and empirical results, building on a historic narrative of opposition to the synthesis. So for instance, the early evo-devo pioneers like Per Albrecht were talking about generative biases in kind of the same way that I talk about them today. Okay, so I'll stop here and take questions. Thank you very much. This is a work in progress. There's a lot more to say. My big challenge in putting this together was to cut it down from like 85 slides to to a reasonable number of slides that I could do in 45 minutes. So thank you again.